Hi everyone. Today in this video let us discuss about the aromatase inhibitors. What are the aromatase inhibitors? Aromatase inhibitors are the anti-cancer agents which are used in the postmenopausal women, and they are particularly used to treat the breast cancer. So these drugs are useful in the treatment of postmenopausal breast cancer. So these drugs are useful in the breast cancer which is hormone responsive. That means the breast cancer which is depending on the estrogen activity. And what are the breast cancer which is in the advanced and metastatic stage? And finally, the cancer which is not responding to the treatment of tamoxifen. Tamoxifen is an estrogen antagonist which is used to treat the both pre as well as postmenopausal breast cancer. So in the postmenopausal woman, if tamoxifen is not sufficient to control the breast cancer, then these aromatase inhibitors can be given. So in all these clinical uses, we can see that these drugs are useful only in the postmenopausal woman. That means they are not preferred in the premenopausal women, and in the premenopausal women we can use the estrogen antagonist like the tamoxifen in order to treat the breast cancer. So today in this video let us see why these aromatase inhibitors are useful only in the postmenopausal women, and what is the role of this enzyme, and how these drugs are going to inhibit the enzymatic activity. First of all, let us see the role of the aromatase enzyme. For many of the steroidal hormones, the cholesterol is the initial precursor. So cholesterol can be converted into various types of steroidal hormones. It can be converted into progestogens as well as androgens and estrogens. And apart from these, it can also be converted into glucocorticoids and mineralocorticoids. But here the estrogens are mainly producing the breast cancer by acting on the estrogen receptors. So what are the drugs used to treat the breast cancer should inhibit the estrogenic activity. During the synthesis of the estrogens from the cholesterol, so many steps are involved, and among them one of the important enzyme is the aromatase enzyme. This aromatase enzyme is responsible for the introduction of the aromatic nature to the steroidal ring A. And androgens like the testosterone can also be converted into estrogens by the same enzyme, aromatase enzyme. Now aromatase inhibitors are going to inhibit the which type of enzyme? Whether they are going to inhibit the synthesis of the estrogens from the cholesterol, otherwise the conversion of the androgens to the estrogens. In order to understand this, now let us see what is the role of this enzyme in premenopausal women as well as postmenopausal women. First of all, let us see what is the role of this aromatase enzyme in the premenopausal women. Before the menopause, estrogens are going to be biosynthesized within the ovaries. One of the important estrogen is the estradiol, which is having the high estrogenic activity. But this estradiol can be converted into other metabolites like the estrone and estriol, which are somewhat less active. So estradiol is very important for the estrogenic activity, which can increase the risk of the breast cancer. So this is the structure of the estradiol, and you can observe within the steroidal nucleus the first ring is having the aromatic ring. Now these estrogens are derived from the cholesterol by several steps, but the main steps are the conversion of the cholesterol into the pregnenolone. This pregnenolone is then converted into androstenedione, and finally this androstenedione is converted into estrogens like the estradiol. So these are the various steps, but let us see at which step this aromatase enzyme is going to be involved. So this is the structure of the cholesterol. This cholesterol is the unique precursor for many of the steroidal hormones. In the estrogen biosynthesis, the cholesterol is initially converted into one of the intermediate, that is the pregnenolone. You can see what is the change from the cholesterol to the pregnenolone. In the pregnenolone, the side chain is having only 2 carbons. So pregnenolone is having the pregnane nucleus, whereas cholesterol is having the cholesterol nucleus. So in this conversion, only the length of the side chain is going to be reduced. So here the aromatase enzyme is not involved. And second step is the conversion of the pregnenolone into the androstenedione. So pregnenolone is having the 2 carbon chain, which is then converted into androstenedione. The androstenedione is not having any chain at the 17th position. So again here the ring A is not converted into aromatic ring, so aromatase enzyme is not involved here. Then the last step is the conversion of the androstenedione into the estrone. Now you can see that the estrone is having the aromatic ring, so here the aromatase enzyme is going to be involved. In this way, in the estrogen biosynthesis, aromatase is going to be involved in the conversion of the androstenedione to the estrone. Then what is the activity of the aromatase inhibitors at this step? Aromatase inhibitors are preferred in the postmenopausal women. They are not used in the premenopausal women. If they are used in the premenopausal women, they can inhibit the estrogen biosynthesis, which lead to the various unwanted side effects. So here aromatase inhibitors are not acting at this step. Now let us see what is the role of this aromatase enzyme in the postmenopausal woman. After the menopause, estrogen biosynthesis is somewhat reduced, so estrogens cannot be biosynthesized from the ovaries, but a small amount of the estrogens can be biosynthesized in the peripheral tissues. For example, at the adipose tissues located on the breast involve the estrogen biosynthesis, but for this estrogen, the androgens are acting like the precursors. So one of the intermediate like the androstenedione can be converted into estrone, otherwise the testosterone can be converted into estradiol. These two steps are going to be mediated by one of the enzyme, aromatase enzyme. Now this is the androstenedione. You can see it is not having any aromatic ring. Now it is going to be converted into estrone, where estrone is having the aromatic ring, and this step is mediated by aromatase enzyme. So here the aromatase inhibitors are going to inhibit this step, thereby they inhibit the synthesis of the estrone from the androstenedione in the postmenopausal woman. Similarly, this is a testosterone, and testosterone can be converted into estradiol by the same enzyme, aromatase enzyme. Again, this step is going to be blocked by aromatase inhibitors such that they are going to inhibit the synthesis of the estradiol from the testosterone. In this way, aromatase inhibitors are going to inhibit the estrogen biosynthesis in the peripheral tissues from the androstenedione as well as the testosterone. Now let us see the different types of aromatase inhibitors. They can be classified into two types. They are steroidal as well as non-steroidal. Among the steroidal, one of the old generation drug is the Exemestane, but nowadays the new generation is the Exemestane. Similarly, in the non-steroidal, we can observe the old generation aminoglutamide, but the new generation drugs like the anastrozole and letrozole. So here non-steroidal drugs are having somewhat less side effects compared with the steroidal agents. And among the steroidal agents nowadays, Exemestane is available for treatment of breast cancer in the postmenopausal woman, and this drug produces an irreversible inhibition of the aromatase enzyme. Anastrozole and letrozole are acting like commutative inhibitors of the aromatase enzyme. And aminoglutamide is one of the non-steroidal agent, but it is a old generation aromatase inhibitor. This drug is going to inhibit the conversion of the cholesterol into the pregnenolone. We have seen that the conversion of the cholesterol and pregnenolone is a common step in many of the steroidal hormone biosynthesis. So by inhibition of this step, aminoglutamide can inhibit the synthesis of the androgens, estrogens, glucocorticoids as well as mineralocorticoids. So this drug is not specific to the estrogen biosynthesis. It can inhibit the biosynthesis of many of the steroidal hormones. And since the glucocorticoids are not synthesized, the cortisol levels are going to fall, which increase the ACTH levels. That's why along with the aminoglutamide, hydrocortisone should be given in order to control the elevated levels of the ACTH. Side effects of the aromatase inhibitors. One of the important side effects of the aromatase inhibitors are the decrease in the bone mineral density. Because estrogens are required to maintain the bone mineral density, when the estrogens are not synthesized, particularly in the postmenopausal woman, the bone mineral density is somewhat reduced. And second one is the increase in the cholesterol levels. Since the estrogens are not synthesized, the cholesterol levels may be increased, so hypercholesteremia may be observed with the aromatase inhibitors. And other side effects like fatigue, dizziness can also be observed with these drugs. Hepatic failure is also observed on long-term treatment of aromatase inhibitors. Similarly, hot flushes, arthralgia and headache are observed with the steroidal aromatase inhibitors like the Exemestane. In this way, aromatase inhibitors are going to inhibit the biosynthesis of the estrogens from the androgens in the postmenopausal woman. Thereby they inhibit the estrogenic activity. That's why these drugs are useful in the treatment of postmenopausal breast cancer, but the breast cancer should be hormone responsive. Then only these drugs can show their therapeutic activity. So that's about the aromatase inhibitors. If you like this video, please subscribe to our channel, share this video with your friends, post your comments in the comment box. Thank you for watching this video.